Exactly how are vaccines made?
Vaccines are biological products, which means they're made from living organisms. This fact makes a huge difference in how they're manufactured. Take safety, for example. At each step of the manufacturing process, extreme care and strict precautions are taken constantly. Production takes place in a controlled atmosphere and absolutely sterile conditions. Anything that enters or exits the production area is carefully monitored. Water and other fluids, even the air in the room, which is constantly checked to ensure it's sterile. To avoid any risk of contamination, production technicians wear high protection clothing. Gowning up is no easy task. In fact, the production agents are specially trained for this. Quality is the next essential ingredient. Manufacturing a vaccine is a complex process involving many different steps, and each step in the cycle is strictly controlled from start to finish. Continuous monitoring along the entire production chain ensures that raw materials, equipment, and manufacturing processes, and naturally the finished products, are of the highest quality. The total amount of time devoted to tests and controls is substantial. More than 70% of the time required to produce a vaccine is spent on these controls. Each and every batch of vaccine that is manufactured must undergo routine testing to ensure purity, efficacy, microbiological security and safety. There are no exceptions. If a batch does not meet the quality criteria, it will not be distributed. For some vaccines, more than 50 different tests may be carried out at various stages of production. As the batches come out of production, samples are taken from each batch and sent to the health authorities. These samples undergo another series of tests. Only after the vaccines have successfully passed all these tests can distribution begin.
Considering that vaccines are going to be injected into a healthy body, so many precautions are understandable. I never imagined making a vaccine could be such a complex process.
It's also a very long process. The total time required to produce a vaccine, if one includes all the controls and checks, ranges from 6 to 22 months. For example, 6 months for an influenza vaccine and 22 months for a polio vaccine.
So how's the production organized?
Production starts with growing viruses or fermenting bacteria. Very small quantities of these microorganisms are caused to multiply in order to harvest extremely large quantities. From just a few cubic millimeters of solution, it is possible to produce enough virus or bacteria to develop millions of doses of vaccines. Each microorganism requires specific conditions to reproduce. Bacteria multiply all by themselves, but they need to be put in the right environment, which is carefully monitored. Viruses are different. They need other living cells, host cells, to be able to reproduce. The first step, therefore, consists of growing huge quantities of these cells and then adding the virus so that it will be able to multiply. Microorganisms are grown in special, complex growth media that may contain up to 30 different ingredients. A wide range of parameters must be taken into account: temperature, pH, oxygen rate, sterility, homogeneity, and so on. Each factor is important. The slightest variation in the culture media may compromise the results. Depending on the vaccine, cell culture takes from 2 days to 3 months. Following harvest, the next step is purification, which eliminates any trace of culture media and any other impurities. Inactivation takes place next. This step is both extremely delicate and critical. Inactivation consists of destroying the pathogen's ability to cause disease while making sure they will still be able to induce an immune response. At the end of this phase, one obtains the very essence of any vaccine, its antigenic valence.
And it's this valence that protects the body.
Precisely. When a vaccine confers protection against a single disease, it is called a monovalent vaccine. It is also possible for one vaccine to protect against several diseases by combining several valences. This involves great scientific and technological expertise. Each valence must remain fully effective, and yet it must not interfere with the other valences. This careful, precise combination takes years of research to perfect. Today, it is possible to combine several valences to offer protection against up to 6 different diseases with a single vaccine.
What happens once this antigenic valence is obtained?
Stabilizers, and in certain cases a preservative, are added. The quantities are extremely small, yet they're sufficient to guarantee that the vaccine will remain stable, potent and effective. Sometimes an adjuvant is also added to enhance the immune response. The next step consists of filling syringes or vials with the vaccine. Certain vaccines, such as the measles, rabies and yellow fever vaccines, are not stable in liquid form. They're put through an additional step to stabilize them. Freeze-drying removes moisture from the frozen vaccine under vacuum conditions and at very low temperatures. The product is now a powder. When it's time for the vaccine to be administered, it will be reconstituted by combining this powder with a diluent. When the filling phase is complete, each syringe and each vial undergoes routine inspection. The quality of both the contents and the container are scrutinized. For this phase of visual inspection, the human eye is aided by electronic cameras that detect and identify even the smallest defect. Final tests are performed by the vaccine manufacturer and then again by the health authorities. The vaccines are then packaged and ready for shipping. Once again, certain precautions are absolutely necessary. From the moment the syringes and vials are filled, up to the time the vaccines are administered to patients, maintaining the cold chain is of the utmost importance. Vaccines are fragile biological products, and their potency can be lost if they're exposed to light or to temperatures that are too warm or too cold. To guarantee that vaccines will offer protection against disease, they must be stored at a constant temperature, between 2 degrees Celsius and 8 degrees Celsius. Cold rooms, refrigerators and isothermal packaging ensure that they will be protected during each phase of transportation and storage. A monitoring device is included in each shipment to ensure the temperature is maintained throughout. Even the slightest variation in temperature is indicated.
Now I understand why manufacturing vaccines is such a challenging job.
It is challenging, yes, but it's also very exciting. At the end of the vaccine production cycle, the lives of millions of people are protected against many different diseases, some of them fatal. Today we have vaccines that offer protection against 26 different diseases, such as tetanus, tuberculosis, polio and rabies. Tomorrow, we will be able to fight other diseases, ones for which a vaccine has not yet been developed, such as dengue fever, hospital-acquired infections, and perhaps one day, malaria and AIDS. Vaccination is one of the most innovative fields of medical research, and it has many more fascinating developments in store for us.